Hi everybody, in previous video you already discussed about some introduction to the first kingdom according to R.H. Whittaker. That one is Monera kingdom. This Monera kingdom included Archaebacteria, Eubacteria, Actinomycetes, Mycoplasma and blue-green algae. This means cyanobacteria. Okay. Archaebacteria are the primitive organisms, okay, and also ancient organisms. These archaebacteria can survive in some extreme conditions. These are also classified into methanogens, halophils, thermoeustophiles. Methanogens, these can survive in the high acidic pH value conditions. This means these can survive in cattle ruminant. Thermoeustophiles, this means these can survive up to 80 degrees centigrade temperature. Halophils can survive in salty areas, high salt pans. Okay, and this way on their habitat wise and also surviving some extreme conditions wise, archaebacteria are categorized into methanogens, halophils and thermoeustophiles. Next one, eubacteria. Eubacteria are the most abundant organisms, most abundant unicellular organisms. Majority of the Monera kingdom is occupied by these organisms. Eubacteria are also live in extreme habitats. Next one, these are first time recognized by the Anton van Leeuwenhoek under his own microscope. It was called them animalcules. Then after Ehrenberg called them bacteria. Bacteria term is introduced by the scientist Ehrenberg. On the basis of their morphological structure, bacteria are furtherly divided into cocci, this means round or spherical bacteria, bacillus, these are rod shaped bacteria. Next one, Vibrio, comma or half helical bacteria. Next one, spirillum, that means these are spiral or complete helical bacteria. Some bacteria can change their shape on the basis of their surrounding atmosphere conditions. These are called pleomorphic bacteria. Pleomorphic bacteria. One of the example, Azotobacter. Azotobacter is the best example to the pleomorphic bacteria. Now in this video, I want to go to discuss briefly about the structure of bacterial cell. Okay, let us discuss about. So this bacterial cell. Okay, bacteria cell is a prokaryotic cell. Okay. That means nucleic acid, nucleic material, that means GNO4, nucleic material is present but is not enveloped by any membrane layer. So, nucleic acid is present, genetic material is present but is not enclosed with membrane. So, nucleus is absent. So, the bacterial cell is one of the example of prokaryotic cell. Okay. The bacterial cell structure, this means prokaryotic cell structure, is distinguished into different types of parts. These are the outer membrane, capsule. This one is the cell wall. Innermost of the cell wall consists cell membrane. It is also known as plasma membrane or cytoplasmic membrane. Okay. The cytoplasmic membrane is surrounded by the cytoplasm of the cell. Okay. The cytoplasm of the cell consists only ribosomes, and remaining cell organelles are completely absent. Only 70 S ribosomes are present in the cytoplasm, and genetic material is freely present in the cytoplasm. It is not enclosed with any membrane. In addition to the nucleic acid, a small circular DNA is present. It is called plasmid. It is highly used in the genetical engineering technology as a vector. Understand? Let us discuss about the first one is the cell wall include glycocalyx, flagella, fimbria, and pili. These are the external appendages. Okay, external structures. What are they? Glycocalyx, flagella, fimbria, and pili. The first one is glycocalyx is a sticky gelatinous material surrounded by the bacteria. It is the outermost layer. And also it is loosely distributed around the cell. It is known as slime layer. Jiguru pora అని చెప్పేసి అంటారు. Okay. If this gelatinous layer, a glycocalyx sticky gelatinous layer, it is loosely distributed around the bacterial cell, it is known as a slime layer. If this sticky gelatinous layer is firmly attached to the cell wall, it is called capsule. Understand? Capsule is made up of polysaccharide and amino acids. Understand? Capsule is made up of polysaccharide and amino acid. This one is the dotted layer is capsule. Capsule is made up of polysaccharide and amino acid. Remember this one. Okay. Presence of this capsule is genetically controlled. క్యాప్సూల్ ఫామ్ అవ్వాలా లేదా అనేది దేని కంట్రోల్లో ఉంటుందంటే జెనెటికల్ కంట్రోల్లో ఉంటుంది. అండర్స్టాండ్? ద క్యాప్సూల్ బ్యాక్టీరియాస్ ఆర్ హైలీ వైరులెంట్, వెరీ డేంజరస్ బ్యాక్టీరియాస్. దేర్ కాజెస్ డిఫరెంట్ టైప్స్ ఆఫ్ డిసీజెస్ బోత్ ఇన్ ప్లాంట్స్ అండ్ యానిమల్స్. అండర్స్టాండ్? Okay, the capsule containing bacteria are highly virulent bacteria. Understand? The next one, flagellas are present in all bacteria except some spherical bacteria called cocci bacteria. These are the non-motile bacteria. Spherical or round-shaped cocci bacteria are the non-motile bacteria. Okay. Except these cocci bacteria, remaining bacteria contain flagellas for their locomotion. Understand? On the basis of the number of flagella, there are classified into following categories. What type of categories? The first one is monotrichous. The monotrichous bacteria are consist only one flagella at the only one polar end of the bacteria. Understand? Only one flagella is present either one end of the polar. This type of bacteria are called monotrichous. Second one, lophotrichous. This means more than one flagellas are present at one polar end of the bacteria. This type of bacteria are called lophotrichous. Next one, amphitrichous. Two or more than two flagellas are present either polar end of the bacteria. Understand? Peritrichous means more than one flagellas are present throughout the surface of the bacteria. This type of bacteria are called peritrichous bacteria. Another last one is atrichous. Flagellas are absent. Flagellas are absent. This type of bacteria are called atrichous. Okay. Basically, on the basis of the presence of flagellas on the surface of the bacteria, they are categorized into monotrichous. A single flagella is present either one polar end of the bacteria. Lophotrichous, more than one flagellas are present one polar end of the bacteria. Amphitrichous, two or more than two flagellas are present either polar end of the bacteria. Peritrichous, this means numerous flagellas are surrounded by the bacterial surface. Atrichous means flagellas are absent. In this way, on the basis of number of flagella, the bacteria are classified into monotrichous, lophotrichous, amphitrichous, peritrichous, and atrichous. Understand? Another one, fimbriae. These are the bristle small-like structures. Small-like structures compared to the flagellum. Okay, it composed of a specialized protein is called pillin. Pillin protein. Flagella is made of a flagellin protein. This fimbriae is made of a pillin protein. Because of this reason, it is called pillis. Pillis are two types, normal pillis and sexual pillis. Okay, they are commonly found in the gram-negative bacteria. We'll discuss about the gram staining technique. Then after, what is the gram-positive and what is the gram-negative bacteria. Okay, there are common in gram-negative bacteria. The next one, they do not help in movement. Do not help to locomotion. Flagellas are help to locomotion, movement of the bacterial cell. But these fimbriae or pillis do not help in movement. They are help to attachment of the host organism or surface. Understand? So these are helpful for the attachment. Bacteria are attached to the host organism with help of these smallest bristly structures are called pillis. Understand? Next one, pillis are normal and sexual. Normal pillis are helpful for attachment to the host organism or surface of the organism. Next one, sexual pillis are helps to genetic material transfer. Genetic material transfer. Suppose F plus and F minus strain bacteria. F plus, F minus. These are the donor cell. These are the recipient cell. These are donor genetic material. These are receiving genetic material. So these are the opposite cells. These are attached with one another with help of the sexual pillis. Sexual pillis. By the fusion of these sexual pillis, a conjugated tube is formed in between the opposite cells. The genetic material is doubled and transferred through these conjugated tube and receiving recipient cell. Then after, both cells are converted into recipient cell, F plus strain cells. This phenomenon is known as conjugation process. In this conjugation process, sexual pillis are help to genetic material transfer. It is known as conjugation process. In this way, at the time of sexual reproduction, at the time of genetic material transfer, at the time of conjugation process, sexual pillis are helpful for the formation of conjugation tube. In this way, the normal pili are helpful for the attachment, and the sexual pili are helpful to the genetic material transfer at conjugation. Understand? Okay, the next one is cell wall. Cell wall of the bacteria. Bacteria contain rigid cell wall. Rigid cell wall, it is a plant character. Cell wall contain character is a plant character. Next one, the cell wall is made up of peptidoglycan, or murin. But archaebacteria consist to pseudomurin. Because of the presence of pseudomurin, archaebacteria can survive in some extreme conditions. Okay. Eubacteria are also can survive in some extreme conditions because of the presence of murin cell wall containing material. Okay, murin is one of the polymer of two sugars, and N-acetyl glutamine based branches are present in each murin. It is a glycosidic bond material of the murin. Okay. Next one. Christian Graham. Who is the scientist? Christian Graham. Christian Graham divided bacteria into two categories by the nature of the cell wall. It is called Gram staining technique. Okay. On the basis of the Gram staining technique by the Christian Graham, bacteria are divided into two categories. What are they? Gram positive and Gram negative. Gram positive and Gram negative techniques. Say this video.
Gram staining. The Gram stain is one of the most usual methods in identifying bacteria. It is an example for differential staining technique. In this procedure, the bacteria is stained and examined under the microscope. Gram stain permits the separation of all bacteria into two large groups, those which retain the primary dye or Gram positive and those that take the color of the counter stain or Gram negative. Procedure. Heat the inoculation loop with burner. Start burning the loop horizontally from its base to vertical position till it becomes red hot. Touch an isolated colony with inoculating loop and swirl it in the drop of water on the slide. If it is broth culture, a loop full of culture can be placed directly on a clean microscopic slide. Now let the smear air dry at room temperature. Now fix the smear using heat by waving the slide over a flame and be careful not to overheat. Flood the slide with crystal violet and let stand for 1 minute. Wash the slide briefly with cold water. Then flood the slide with Gram's iron, let stand for 1 minute, wash off with water. Decolorize until the water flows colorlessly from the slide. Now flood the slide with safranin, let stand for 30 seconds, wash off with water now. Blow the slide dry with bulbous paper. Now examine the slide under the microscope for Gram reaction in 100x oil immersion objective lens. Now we can observe that Gram positive organisms appear purple in color while Gram negative organisms appear pink in color. Now some tips for the successful results. Only perform Gram stains on fresh cultures or log phase culture after 24-hour incubation. Be sure to stop the application of decolorizer immediately after it runs clear to avoid over decolorizing.
Another one, cell membrane. Okay. The cell membrane is present innermost of the cell wall and it is surrounded by the cytoplasm of the bacterial cell, and it is made up of, it is consist of phospholipids and also proteins. Proteins are 60% and phospholipids are 40%. Next one, the cell membrane is a selective permeable. Only some ions are pass through this membrane, allow through this membrane. So these are act as a selective permeable membrane and also helps in osmosis regulation, osmosis, and also helps in facilitated diffusion. Only some ions, only some cations and anions are passed through these membrane. Facilitated diffusion, only selectable ions are passed through this membrane. Hence, it is also helps in facilitated diffusion process. Understand? Next one, cell membrane forms some invaginations. These are the red colored membrane is cell membrane. This red colored membrane. Okay. This membrane also shows some invergations. These are the invergations. These invergations are called mesosomes. Mesosomes. These mesosomes are helpful for mainly these cell membranes are so some invergations are called mesosomes. These are also consist of some enzymes responsible for DNA replication and also binary fission. Okay, especially mesosomes are involved in the DNA replication, binary fission. By the presence of respiratory enzymes, there are also performed respiratory process. Understand? This one is a very important structure in the cell membrane. It is known as mesosomes. Understand? The next one, cytoplasm. The cytoplasm. The innermost of the cell membrane, the living solution is known as cytoplasm. This cytoplasm surrounded by the cell membrane, and cytoplasm is a living solution. It consists of different types of components like micronutrients, macronutrients, and also some ions and ribosomes. These are the cell organelles. These ribosomes are the 70S type. Largest subunit is 50S, smallest subunit is 30S. Understand? Next one, some food storage granules are also present in this cytoplasm. They are called glycogen. This means the food is stored in the form of glycogen. Another bodies are also present in the cytoplasm. They are called PHB. This means polybeta hydroxy butyric acids. Beta hydroxy butyric acid, PHB granules are also present in the cytoplasm. Some inclusion bodies are also present in the cytoplasm. These inclusion bodies are the non-living structural materials. Some non-living structural materials are also present in the cytoplasm of the bacteria. These are called inclusion bodies. Next one, polysomes. Some polysomes are also present in the cytoplasm. At the time of protein synthesis, 70S type of ribosomes are attached to mRNA. Okay. So many member of 70S ribosomes are attached to the mRNA at the time of protein synthesis. The structures are known as polysomes. Okay. The polysomes are also there in the cytoplasm of the bacterial cell at the time of protein synthesis. Next one, gas vacuoles. Sap vacuoles are absent. Remember this one. Sap vacuoles are absent, but gas vacuoles are present. Gas vacuoles are present. Okay, these are the membrane bounded and also protect from the radiation and help in the beyonds. బ్యాక్టీరియాస్ అనేవి తేలియాడడానికి ఉపయోగపడుతుంటాయి అండ్ ఆల్సో ప్రొటెక్టెడ్ ఫ్రమ్ ద రేడియేషన్ రేడియేషన్స్ అనేవి తట్టుకొని సర్వే అవ్వడానికి దీస్ గ్యాస్ వాక్యూల్స్ ఆర్ ఆల్సో హెల్ప్ఫుల్ అండ్ ఆల్సో దీస్ ఆర్ ద membrane bounded structures, gas vacuoles. Okay, membrane bounded structures. The next one, magnetosomes. Some magnetosomes are also present in the cytoplasm of the prokaryotic organisms in the Monera species. These magnetosomes are especially found in Magnetotacticum and also Aquaspirillum species. Aquaspirillum species especially found in magnetosomes. Okay, this one is the cytoplasm of the bacterial cell. Understand? The next one, nucleoid. It is also called genophore. This green colored portion is the genophore. It is also known as nucleoid. It is a single circular DNA. Nucleoid is a single circular DNA. It is not enclosed with any nuclear membrane. Okay. Next one, it is made up of nucleic acid and also proteins. So these are the nucleoproteins. Next one, it is like histone proteins. Okay, normal గా ఉండే DNA లో eukaryotic DNA లో histone proteins are present, but in this nucleoid single circular DNA lacks histone proteins. Hence it is known as naked DNA or genophore. It is also called genophore or naked DNA or single circular DNA or bacterial chromosome. Bacterial chromosome anna, single circular DNA anna, naked DNA anna, genophore anna okati. Understand? Next one, in addition to this genophore, in addition to this bacterial chromosome, in addition to this naked DNA, in addition to this single circular DNA, a small extra chromosomal circular double stranded DNA is present. A small extra chromosomal double stranded circular DNA is present. This one is a circular DNA. It is now called plasmid, not plastid, plasmid. Understand? This plasmid is a self-replicated DNA. So it is highly used in genetical engineering technology as a vector. Okay, the wanted DNA transfer through these wanted host organism with help of these plasmid because it is a self-replicated structure. Understand? Next one, episomes are also present in some species. Okay, these episomes are replicated with genophore, with entrapted. Okay, these plasmids are entrapted with the genophore and are replicated. These are also known as episomes. Episomes are nothing but plasmids are entrapted to genophore. Plasmids are entrapted to genophore and replicated with the genophore. They are called episomes. zones. These are the complete structure, briefly discussing structure of the bacterial cell. Understand?